Introduction and structure of DNA. Introduction. Nucleic acids are building blocks of organisms which are biological in nature. They are of two types, namely DNA and RNA. DNA stands for Deoxyribonucleic acid and RNA stands for ribonucleic acid. Nucleotide. Nucleic acids are polymers of nucleotides. Each nucleotide is made up of pentose sugar, phosphate, and nitrogen base. Pentose sugar. Pentose sugar is a 5-carbon sugar in the form of ribose sugar of RNA and deoxyribose sugar of DNA. Phosphate. Phosphate molecules are attached to the adjacent pentose sugar, alternatively at their 5th and 3rd carbon atoms, by phosphodiester bonds. Nitrogen base. Nitrogen bases are heterocyclic compounds made of carbon and nitrogen. They are of two types, namely purines and pyrimidines. Purines are dicyclic compounds with 2 CN rings. Different types of purines are adenine and guanine. Pyrimidines are monocyclic compounds having 1 CN ring. Different types of pyrimidines are cytosine, thymine, uracil. Cytosine and thymine are found in DNA, whereas cytosine and uracil are found in RNA. Nucleoside. It is the combination of a nitrogen base and a pentose sugar. A nucleoside, along with the phosphate, constitutes a nucleotide. The DNA. A long polymer of deoxyribonucleotides is called DNA. DNA was first identified by Friedrich Miescher in 1869 and was named as nuclein. The two types of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA. Deoxyribonucleic acid is the genetic material carried from parent to offspring. DNA is found in the chromosomes of the cell. Some amount of DNA is present in the nucleolus, mitochondria, chloroplast, and in the cytoplasm of bacteria. Double helical model of DNA was proposed by Watson and Crick. DNA is a double helical structure comprising of 2 strands antiparallel to each other, that is 5' to 3', 3' to 5'. Both the strands are held together by hydrogen bonds formed between purines and pyrimidines of the strands. According to Erwin Chargaff, The base ratio may vary from species to species, but it is constant for a given species. Therefore, the amount of adenine is always equal to amount of thymine. The amount of guanine is equal to cytosine. The two strands of DNA are coiled to form a double helical structure at an angle of 360 degrees. Each turn of a DNA helix has one minor groove and one major groove. The length of each turn is 34 angstrom units, or 3.4 nanometers, accommodating 10 base pairs or 20 nucleotides. The internucleotide distance is 3.4 angstrom units, or 0.34 nanometers. DNA has a very high molecular weight. It can absorb UV light at a wavelength of 260 nanometers. It gets denatured when heated up to 70 degrees centigrade. Low salt concentrations and high pH values in a cell can also denature the DNA.